Hello. Hello.
Hello, everybody. Welcome to our workshop. I think we'll just wait 3 more minutes until we start.
Hello, Ria. Hello.
Yeah, I'm ready.
Okay, I think we can start now. I'm really happy to see so many of you joining this workshop. So my name is Ria Kubialka. Let me start my video for now. Hello, everybody. I think most of you know me already. Yes, I'm a postdoctoral researcher at the University of Leipzig in Germany. And I'm today, will guide you through the different sequencing platforms and their uses for Oxford Nanopore. And here with me are my colleagues Ariana Ceruti and Prakash Ghosh, also members of the University of Leipzig. I think they will shortly introduce themselves when they are going to present their different topics. And so today we are going to explore with you a little bit the different sequencing platforms. Of course, there are a lot of them outside, but we will today focus on 3 of them, which we use a lot here at the university and which we find quite useful for our needs. So I will turn off my video to make it a bit easier and to show a good connection. So let me first start. Just a second. Yeah. So let me quickly give you a brief overview about the method itself. So nanopore sequencing is a sequencing method which can be used in a really, really small device, which makes it really handy for the use at field settings or at point of need, because in the recent years, I would say the sequencing was bound to a well-equipped laboratory and to huge machines, and this nanopore sequencing really enabled the sequencing going directly into a field. For example, here in the use of a mobile suitcase laboratory, because you can do the sequencing with the help of this little device here you can see on the left-hand side of the suitcase. And just give me a second. I need to stop something here. Yeah, okay. So just, okay, sorry, there was an echo. I needed to switch off the sound. So just give you a quick overview about the method itself. So inside of the device, you use a flow cell. And in this flow cell, you've got a membrane with an electrical current. So inside of this membrane, you can see those pores. Let me see if you can. Yeah, no, I think I can't show my arrow here, but these pores are inside of the membrane, and when you add your sample DNA, so it must be extracted DNA or RNA, the DNA will flow through the pore, through the membrane, and change the current of this membrane. And this change in currency can be detected and is specific for each base that slides through the pore. So depending on the intensity of this current, you can then translate the whole measurement of the current into the specific bases. Let me explain it to you again in a short video. I think that make it a little bit more easy. So here you can see this pore inside of a membrane, and then you've got different proteins which are keeping the pore open, and then as well helping the DNA bind to or find this pore with this adapter here, which is also a protein. And then this adapter leads to the sliding through of the DNA through this pore. And as you have many different DNA strands inside of your sample, each sample gets connected with this adapter and can be slide through this pore. And here you can see the current changes while the bases are slipping through the pore. So in the end, you've got thousands of bases here, and each is detected via this change of currency. And whenever one DNA snippet, I would say, or DNA piece is done or has ended, the DNA will just free the pore because it will go away from the protein inside of the membrane. So the pore is again free for the next DNA. And the devices which are developed by Oxford Nanopore are called MinION. So it's not the minion here, but it has a similar size, I would say. So it's called MinION. I don't want to go into a deep explanation of the sequencing kits you can use, but I just want to highlight one kit we are frequently using here at the lab, which is called the Rapid Barcoding Kit. And here is the best about this kit is that you can add to each of your samples a barcode transposome complex, and this leads to the marking of each sample. So each sample gets a specific barcode, which is then tagged to the end of your DNA. So the good thing here is that if you have, for example, 50 samples, you can add this barcode to each sample. So the DNA in this sample gets the same tag. And then in the end, you can pool all of your samples together, because in the end, you can still tell which sample or which DNA belongs to which sample, because they have the specific barcode. And these barcodes are numbered from 1 to 96, depending on the kit you are using. So you can pool up to 96 samples in one sequencing step, which makes it a lot more easy to do the whole sequencing. And then there are, of course, also kits for RNA. it depends also always on your needs, which kits are best for you. So here you can see example of a flow cell which we are using for sequencing. The most important parts of this flow cell is, first of all, here the priming port where you add the buffer mix, and then you've got here the spot on port where you add your sample in the end. And you can use the flow cell once, or you can even wash it and then use it again. So just to give you some general information about the devices which you can use for field use, there was the MK1C, which is not sold anymore. Maybe some of you have still have it in the lab, so you can still use it for, I think the next 2 years at least. But it's not sold anymore, so you should sooner or later swift to one of the other devices. So you've got the MK1B, which you can connect to your laptop or computer. And then you've got the new device, which is the MK1D, which is also quite small. It's comparable to the MK1B, but you can also get it together with the tablet, so you don't need your own laptop or computer anymore. Then maybe just two general informations. If you have a MinNow software version, which is older than the 24.06, you cannot use it anymore. So it's expired. It expired already on January 4th. So you really have to take care that you've got the latest update. And then maybe a thing for all the Epitomi users here, the Epitomi agent software expired last year already and cannot be used anymore. So you have to download a new software, which is just called Epitomi now. But to we will explain that later on, or my colleague will. So just to give you a quick comparison of the different sequencing platforms, we are going to talk to you today about the Epitomi software, the Genius software, and the CZID software. The BugSec we are not going to explain further today because it is quite an expensive software to use. I mean, you've got the free trial for 3 analysis runs, but then afterwards it's quite expensive, and it's always depending highly on your needs. So they are quite focused on your needs, so you can talk to them directly, and then you get an offer from them depending on how many samples you want to sequence and everything. And they've got a really good compliance and really good ID behind this last program, the BugSec. But for the ones who are not using sequencing too frequently and where you don't have a lot of money available, I wouldn't recommend this platform for you. And the other 3 are quite comparable. The results can be found within up to 1 hour, I would say. The CZID is the quickest one here, and it also has the most user-friendly platform, I would say. And also an advantage of the ZZID is that it's free. The Epitomi is free for Oxford Nanopore technology users, and the Genius is costing a few US dollars for one academic user. And maybe another thing we should highlight here is that the Genius one is the only one which you can use for offline data usage and storage. So for all of the other programs, you need Wi-Fi access or internet access. But let me now hand over to my colleague Prakash Ghosh, who will now give you a short insight into the EPI2Me program. Yes, Prakash, please go ahead.
Thank you, Ria, for very nice introduction. Nanopore sequencing. Hi, everyone. So I'm going to present the EPI2Me. So before going to the presentation, I would like to give a brief introduction about me. So I've been working as a PhD student at University of Leipzig, and I'm also working with Ria and Ari. So let's have a brief primer on the EPI2Me. So EPI2Me is a universal platform to analyze the long-read sequences so that we get from Nanopore sequencing. So it has versatile applications. You can use it for human genetics and genome assembly, metagenomics, and to detect infectious diseases, and you can go for target sequencing, and also the detection of antimicrobial resistant genes. So EPI2Me has many workflows. So it has more than 15 workflows right now. So you can do whatever you want according to your aim and what is the objective of your study. So I would like to give a short brief why we are interested in the workflow that is a bacterial whole genome workflow. Because our aim is to detect the antimicrobial resistant genes in the clinical isolates. So right now, the antimicrobial resistance is a huge problem globally. And we know the major driver of this antimicrobial resistance is the overuse or inappropriate use of the antibiotics. And it's mostly happening in the low middle income countries and in Africa, in Asia mostly. So you know, the gold standard for detecting the antimicrobial resistance is the AST or the culture-based methods, which are very slow and it's tedious, and you need a use laboratory data. So with the advent of nanopore sequencing, you can detect the antimicrobial resistant genes at the remote settings. So now we have the sequencing, but we must need a portable or the real-time analyzing system, because the expertise in bioinformatics is very limited in low research settings. So here comes the EPI2Me, so which can be utilized to analyze data real time, even from the low research setting. And that's how we can get more information regarding the antimicrobial resistant genes, and we can monitor easily even in the remote settings. And because right now we are getting all the information from the high income countries regarding the antimicrobial resistance. So if we'd like to capture more data, and then we can design the interventions, and we can go for more surveillance from the low research settings. So that is the motivation of our study, and therefore we are going to share our experience, how this new workflow is working, and so far what we have done. So this workflow, the whole genome bacterial whole genome workflow, so it can be done locally or it also cloud-based. So if you would like to do it locally, then you have to have particular configuration in your system. Like if you have to have a very good device, and you can see what are the minimum requirements to perform the analysis if you do it locally. And if you do remotely, then you have to have at least 3 threads in your CPU, and also you need a memory. At least the RAM should be 8 GB. So, and if you look at the turnaround time, like what is the time required for particular analysis for each of the samples, it will take around 20 to 40 minutes and even more if you have a very good coverage of your genome. So you can see the vignette of the software after downloading the software. Earlier we used the Epitome agent. I think many of you have already used this software before. So that is the new look of this software. So you can find the workflows, different workflows here, and you can choose as you wish, like depending on your objective of the study. So if you get into the workflow for bacterial genomes, you will find that these two options. You can run your sequences locally, and you can run even through the cloud. So here are the options, like if you have your sequence, then you can input your sequence in particular, like you have two options. If you would like to submit your FASTQ files, and also there is another options to submit your BAM files after following the alignment. So you can submit the raw FASTQ files for analyzing your data. So I'll show you the real time how you can submit your files, and you can get through the analysis, and also how you can retrieve your report. So before going into the real-time demo of the data, how you can analyze, you can submit, and also you can retrieve the report. So I'd like to give a brief what is happening in the whole system, like in the whole workflow, what we'll see. So after the nanopore sequencing, we usually get the raw reads. So here I would like to give one piece of information that is really important. So there are 3 types. I think many of you already know how to do the sequence the data, and there are 3 different options for the base calling. So one is fast, one is high accuracy, and a superior high accuracy base calling system. So if you would like to go for assembly, then you have to have the more accurate base calling model. So therefore, your assembly will be more accurate. So therefore, you have to have the raw reads that must have the high accuracy, and even you can have the super high accuracy. That both are okay. So now after having the raw reads, there are certain softwares. One is a FastCat and one is BAMStat. So what they will do, so they will see what is the accuracy level, what is the quality of the reads, and what is the length of your reads. So you can see you can see here the graph showing the read quality and the length of the reads. And after that, so based on the reads, there is a fly assembler that will make the contigs. And we'll get these contigs from the overlapping the raw reads that we get from the individual isolates, and then it will make a draft assembly with a fly tool, and then there will be another software that is called Medaka that is only for long reads. So these all the tools are made for the long reads, and then after the polishing of the assembly, so we'll get more accurate assemblies, and then the assembly will be submitted to different databases. Like there are two different tools. One is called Procta and another is also ResFinder. In the ResFinder, if you submit, then you will get the AMR genes. And there is another tool that is called Procta. So that is for annotation. So you'll get the information of the different genes. And if you like do any work on a functional genomics, like the non-coding regions, coding regions, and which the function of the genes, and we'd like to predict new genes. So that is the function of this research. So our focus is ResFinder because we'd like to detect the AMR genes. So here are the important links we'll share, so that will guide you through like how we can analyze yourself and you can get more information. So I think now you can see the workflows. There are different workflows here. So I'm going to select the bacterial genomes. So you can see there are both options for run locally and run in cloud. So I'll go for the run in cloud option. And there here you can see so there are input parameters, so you can submit your raw files. So there are basically three options to select a path. Like if you would like to submit a single FASTA-Q file, so in that case you have to select the specific file, and if you have just one sample, like one barcode, then you have to select that particular folder. And if you have like 10 samples with 10 different barcodes, then you can just select the folder that containing all of the barcodes. So like, so I have many samples. So if you go for the multiplexing, so you can just select the folder, and that's it. And here you will find there are different parameters. So that is for reference. So we'll go for the de novo assembly. We'll not doing any reference-based assembly. So therefore, these options are not for us. And so there are also options for unclassified reads. Like if you have a cutoff, like so usually in antimicrobial resistance gene detection, we usually put a cutoff like a 1,000 base pair for the reads. So because the reads below the 1,000 base pair, those are faulty reads and they're more those reads are incorrect and they have more errors, so they will be considered the unclassified reads. So we'll not consider those. So here you will get also the sample options. So if you would like to put any different, like if you would like to label the particular samples, like which might be corresponding to a particular the samples with barcodes. Let's say barcode 1, you can give a name of this particular sample. So in that case, you have to put a CSV file there. So it's optional. If you would like to give, you can add here, or otherwise you can usually get the output of your result. So there is another option, that is isolate options. So here, if you would like to see, so here you can see, like if you would like to see them, what is the multi-local sequencing type, if you'd like to see what is the strain of your bacteria, and also if you'd like to see what is the antibacterial resistance profile in your bacteria. So in that case, you have to go for it. Otherwise, it will show just some information regarding the annotation. And here, to specific information, those are very important. So basically, in ResFinder, so if you would like to predict the gene in your bacteria, so in that case, you have to go for the cutoff identity and the cutoff coverage for this the gene that you are going to predict. So in your sample, if there is any like a gene and it will be matched with the reference in the ResFinder, and if it covers 60%, then it will like pop up in your report. And if and also if it like identity of your particular gene is 80% with the reference one, then it will come out. Yeah, sorry, it will come out in your report. So these two informations are very important. So these are by default. You can change if you would like to have more accurate result. So here you can have the advanced options. So earlier, the versions of the epitome, those are you would get the by default options, like, but here you can like you can change the programs, and there you will find different options here where you can like you can edit like it depends on your objective, like what types of data you would like to have in the output. So you can change different, like if you would like to go for the reference assembly, so in that case you must have this the genome size of your bacteria. So if it's not your objective to see what is the location of the genes in the bacterial genomes, and if you would like to know what is the location of your like the gene in the plasmid, so in that case you have to play with the different advanced options. And if you'd like to have more accurate assembly, in that case you can play with it. And there is another option that is I have already mentioned, that is the cutoff that will be removed from the analysis. So the reads below 1000 base pair, that will be considered as the unclassified. But if you like to have the data from those small reads, then you can like change your cutoff. So here you can see the threads, like the what is the number of threads in your system. So you have to put like the information of your CPU. So that's how you can like use this platform and I think it's if you run the cloud-based analysis, though, in that case, the minimum is 3 thread. So you must put it at 3. And if you have more, then you can you can increase the thread. And then you can go for the, so we don't have the human data because we sequence the cultured bacterial isolate. So therefore, we are sure that we don't have any human data in our sequence. So it will take time. So here are 3 different options. So here's the name of your run. Here is the details. Here is the reports you'll get. So now I'm going to share one of the reports. How does it look? Okay. So if you run your samples, then your analysis will be in the cloud for a long time. So I don't know like how long they will keep your analysis. So, but so I have my analysis and I can share my report. So that is basically the test run that you will get from the Epitome. So that is the test file you can have and you can run it and see like how does it look. So initially you will get the quality of the rate and the length of the read and what is the coverage of your reads. And so here, usually when you run the sequencing in Nanopore, so usually we put the Fed score is around 9 or 7. It depends on which type of work you would like to do. If you would like to detect any new pathogen, like in outbreak situation, then Fed score is 7 is fine. But if you'd like to go for the functional genomics or something else, so in that case, you have to increase your Fed score. So here you can see that the median is around 8, and the read, you can see the read length, and we removed all the reads, so that is less than 1,000. So here you can see the contigs, individual contigs, and what is the coverage for each of the contigs. So here you can see the contig coverage is or it's not coming here, but I had the report. So the country coverage is around 34. In our previous report, I got it. So regarding the coverage, like usually the bacterial genome size is 5 megabase. So if you would like to go for the functional annotation, you have to have the coverage more than 50x. So here we have less than 50x. That is like 10x is good for detection of the antimicrobial resistant. So since our objective is to detect the antimicrobial resistant genes, so that is no problem. But if you go for functional annotation, in that case, you have to have the coverage more than 50x. So if your genome size is 5 megabase, in that case, you have to have the read around 250 megabase. That means 50 times of your genome size. So here you can see the gene information of the genes, which are like corresponding to individual contigs. And the most important that is the resistant genes. So this was a bacterial genome. So here you can see the particular antimicrobial resistant genes that those are being detected, and at the same time you can see the particular mutations that are related to individual resistant gene, and you can see the resistant genes have been detected against the aminoglycosides, and you can see the identity score and also the coverage for this particular predicted genes. And you can see what is the strain of this particular isolate. So here you can see they have given the data on MLSP. So it has the bacterial strain based on the housekeeping genes or particular sequences. So here you can see the versions of the different tools and softwares that have been used for this analysis. And you can save the data. So there is options here. So you can save the data. And you can have you can save all the files after the analysis. So you will have that options as well. So these are more or less the analysis outcome looks like this, and you can use this report for your own work. And just one information, like the one question might be regarding what is the accuracy of this prediction. So based on the previous studies, the accuracy of this prediction is more than 90%. Because the earlier pipeline that was ARM pipeline, that was more than 90%. So the accuracy of this current pipeline is yet to be explored, but we are expecting it should be more than 90%. So I'm stopping here, so I can see. Yeah, I'm expecting questions from Ali Raza.
Hello, Dr. Prakash. Are you hearing me?
Yeah, we can hear you.
Okay, Dr. Prakash, my first question is that why quality score or Ferret score of 7 to 10 are acceptable in case of long read from Oxford Nanopore? Is because if 1000 bases, 1000 nucleotide are wrong in 100 bases or nucleotide are wrong in 1000 bases, then is it good to predict any bacteria or gene?
That is a very good question. So, like if you would like to go for any work on functional genomics or annotation, in that case, you will have to go for the hybrid assembly because you cannot cover the whole genome. So you must have to have both the long reads and the short reads because you cannot have the perfect genome in that case. So if your Q score is 30, then that will be best. But in most cases, we usually detect the pathogen, new pathogen in the real time. So since the nanopore sequencing system, it's the speed of the base calling is really fast. So in that case, that is the I think it's really difficult to overcome the error rate. So I think that is the limitations of the system, but still they're working. So if you use the high accuracy base calling, in that case, you can overcome some of the limitations and you can find your desired result.
Okay. Can you tell me high error rate base calling software that can correct this error?
Sorry, could you repeat your question?
Like you said, if we use high error rate base calling software, we can identify correct pathogen or genes. Do you know any good base calling software?
No, like in the epitomy, there is a default models. Like for the base calling, there are 3 models you can select. One is fast, one is high accuracy, and another is super high accuracy. So there are 3 different models. So you can select as you wish. Like if you'd like to detect any pathogen in an outbreak situation, you have to go for fast one because you have to have the result very fast. But if you would like to go for the annotations or like a functional genomic analysis, in that case you have to go for high accuracy model. So you have this system already in the nanopore, so you don't need another software. But for the assembly corrections, there are different softwares like nanopore system. They have the Medaka, and there are other softwares that you can use to correct to get more accurate alignment and more accurate assembly.
Okay, thank you so much. But if we use a super high accuracy option of the epitomy, then what score does it give to us about our FASTA file? Like 30 for Ferret score, 20 Ferret score?
No. In our experience, we got it around 15.
50?
Yeah, 15. More than 10. You can get more than 10.
Then that is very good. If you know the software behind this super accuracy, that is very helpful for me because I analyzed data using Linux system and I generated my own pipeline and bioinformatics tool. So I didn't know this point. That's why I am asking about the software behind the super accuracy option.
Yeah, if you would like to go for any hybrid assembly, in that case, you must go for the hybrid model. Otherwise, you cannot correct or you can get more accurate assembly. So, but in recent publications, based on some people they showed that if you go for any hybrid assembly, in that case, both high accuracy or super high accuracy, both work the same. So you can choose any one of these here.
Okay, and sorry, second question. Can you tell me what should be our coverage depth or sequencing depth for our metagenomic data to do assembly? for this metagenomic data? What should be the coverage depth?
I can give you the information regarding the isolates. Like if you go for bacterial, like if you would like to analyze any particular isolate or bacterial genome, in that case, 100x is the desired if you go for the functional genomics analysis. So, but for for nanopore sequencing, it's very difficult. In that case, it really depends on how many samples you would like to run. You like to sequence in a particular run, say, if you would like to have more than 50x coverage in a specific run, you have to have only like 20 samples in a flow cell. So it also depends on the chemistry of the flow cell. If you use the most latest version, 10.4, yeah, 10.4.1. So in that case, if your genome size is 5 megabytes, so if you like to have the coverage more than 50x, so you have to run only 20 or 25 samples. So it really depends on your question, like what you really want.
Dr. Prakash, according to my literature research, I didn't found any exact figure for the sequencing depth for metagenomic data using Oxford Nanopore technology. If you read any paper regarding the sequence depth that is essential for metagenomic data obtained from Oxford Nanopore technology, you can tell me it is very helpful for. Yes.
Yeah, I didn't find that information, but if you'd like to go for metagenome assembled genome, in that case, if you would like to go for MAGS, so in that case, the coverage is really important. So I think it would be helpful if you go to some of the literature that is on MAGS, yeah, metagenome assembly. In that case, the coverage should be really.
Yeah, I try to do assembly using Fly, as you mentioned in earlier, Fly, but I use MetaFly because my data is metagenomic. But MetaFly also didn't work. He didn't even give me contigs. Before contigs, it gave like disjoint read. I got this thing, but even Fly doesn't work on this sequencing.
I think you can try another, like you can go for MetaXpress.
Okay, Dr. Prakash,
Yeah.
Thank you so much for your answer.
Thank you. So we can see Shubhajit Boshu, so you can ask your question.
Prakash, thanks for the wonderful presentation. Just have a few questions. How much important is the base calling parameter? And one of my major queries is this, that when we are getting or extracting DNA from, let us say, from the environmental samples, how do we kind of ascertain that we have the depth of the sequencing to ascertain that we can, I think it's a continuation of the previous question, but how do we kind of generate enough data for to assemble, metagenomically assemble genomes, MAGs? What we do about it? Is it about the system? Is it about the base calling parameter? Is it about the extraction technique? So, what is most important that we must think about this?
So, thank you for your question. So, I would like to say that it depends on your question. The regarding on your first question, if you would like to increase the accuracy of the base calling, so if you'd like to detect the pathogens in real time, like in an outbreak situation, that is your objective should be like you have to detect very early. But if you'd like to see what is the variant, what is the strain, and you would like to go for work on functional genomics, you would like to see what is the SNPs. So in that case, you really need more accurate data. In that case, you have to go for like a high accuracy, like a Q score should be more than 10. That will be better. So I think if you change the, we don't have many options. If we use the nanopore sequencing, just you can change the different models, and if you can increase the credit score. So that you can do as per your object.
So it depends on the quality of the DNA.
Sorry.
So it may depend on the quality of the DNA that we extract.
So you
It may
you
depend-
you were
it may
asking the
depend on
second.
the quality. I'm asking that does it then we have to work on the methodology part of the extraction, that it may depend on the quality of the DNA. For example, we are let us say extracting DNA from the water or from the sediment or from the soil.
Mhm.
You know, different applications, of course, but this is what we're interested in. And what it means is that means that we have to work on the quality of the DNA that is getting extracted. Because if we want to go to the direction of MAGs, metagenomic ascending genomes, I think
Yeah.
the amount of you know, the data which is
Yeah, I'm coming into your second question.
Yeah.
So so far I have went through some of the new articles. Like you can go for some DNA enrichment methods, which will help. Like if you have like a very little biomass, like how you can increase the quality of the DNA. So how we can get more DNA. So in that case, it might help you. So there are different DNA enrichment procedures. So you can adapt according to your study and what is the samples you are using in your study.
And my last question is regarding this base calling, which is a little bit puzzling. It's a new technology. It's very fascinating, and it's also very interesting because we can take it to the field. The point is that you know, between this fast base calling and the other options, I mean, for the construction, let us say, even if we enrich some sample with some microbiome, let us say, I mean, what may be the, I mean, is it the same? You highlighted a system requirement initially, but do you want to go higher, or we want to go so is it really the system requirement, or is it really regarding the software, specific, something that is happening? Regarding specifically, because if we take it to the field, we are carrying, let us say, a laptop. So how do you understand my point? How do I make sure that it is going to, work?
I think again, the question is like, what do you really would like to do? So with this sequence,
Let us say
with the long list, yeah. So
yeah.
So I think still, if you go for metagenome assembled genome, so MAGS, I think the work is very limited into some, like it requires like a high facility. You cannot go for MAGS in remote settings, and that is not the objective. I think the purpose, the nanopore sequencing, it can serve towards the democratization of the sequencing. You know, like which information we really require, which are really important in the context. So MAGS is not always like what I should say, like it's not that important compared to detection of the antimicrobial resistance. We really need the information that has the clinical relevance. So we have to work on that. So I think in that case,
yeah, I think we have to work on that, and I think let me bring it down to a different diluted level. Let us say we just want to know from this sample, there are hundreds of so many prokaryotic, eukaryotic species, something phenomena, whatever. So if we want to go for the information, even for that purpose, what do you recommend? Which may be the best way to optimize this method? Starting from the extraction, yeah.
I think we have to wait until the Oxford Nanopore sequencing, they add another workflow for metagenome assays, something like that. So
Yeah.
still
yeah, with this workflow, we can only do limited
Okay.
works regarding the bacterial genome.
Okay, thank you. Please carry on, and thanks for the very nice presentation. Thank you.
Thank you.
Okay, if there are not any more questions for the EP2Me part, I will just go on with the Chan Zuckerberg infectious disease platform. I hope everyone can see my screen now. So the Chan Zuckerberg, or short called CZID, is a software developed by Chan and Mark Zuckerberg, or by the Chan and Mark Zuckerberg Initiative, and it's a platform for analyzing new generation sequencing data to detect infectious disease agents. So it's mainly for pathogen detection, outbreak tracking, also AMR monitoring, and zoonotic research. And to give you a short overview about the pros and the cons of this platform. So first of all, it's free, which is a really huge advantage over the other platforms we've presented here. Then it's really user-friendly. So I would say this platform is the one which is most straightforward. And if you don't have any bioinformatic background, this one is the one which is easy to use because it has a lot of default settings. So a lot of filters which you don't have to change or something, because it's just set by default, and they make sure that the quality of your DNA is good enough to get good results. Then it's open sourced and it's cloud-based, which is an advantage because you don't need a huge or a really good computer and huge data processing computer. But at the same time, it's also a disadvantage because there are always like privacy concerns about all cloud-based systems, basically, because there can be concerns about third-party handling, and also the compliance with privacy regulations may arise. So you always have to be really, really careful on, like, if you have really sensitive data, you have to think yourself if you really want to use this platform or not. The uploaded data is encrypted, but the access controls ensure that only authorized users can view or analyze your data. I will show you in a minute when we do the analyzing on that platform how it looks exactly, but you can choose between public analyzing and private ones, and I would recommend to always use a private one if you don't want to share it with the whole Mark Zuckerberg community. So then talking about the limitations of the Zuckerberg platform, there are only limited analysis types for nanopore sequencing. So there are 4 different types for the Illumina sequencing, but only 2 types for nanopore sequencing, which is very limited in this case. Then you've got the internet dependence, which is the same like the EP2Me platform. So you need internet access to do your analyzing. And then you have a dependency on database updates. So you are really depending on the platform itself, that it uses the recent updates. If they don't update it frequently, you won't have the latest databases which are used for your analysis. And then the last point is that it's not comprehensive for all cases. So if you're looking only for really, really rare pathogens or rare microbes you want to detect, this platform is not the best one to use because it's biased. It is referring more or less to the or using databases with the most common sequences. So if you want to detect really rare sequences, this is not optimal for you. So let me now guide you through analysis using this this program. I will stop sharing my screen for now, and share with you now the setup. If you do the just a second. Here we go. So when you go to the CZID website, you have to log in first. So I will quickly do that for my account. As I said before, it is free to sign up here. So yeah, it should be really easy for you, I think. Can you see, do you see my presentation or the Mark Zuckerberg? Nothing. Okay, perfect. Sorry, I was just confused about the presentation mode here. So, okay, when you open your account, you can see your recent projects here. But I will quickly go from here, my data, to the upload on the right-hand side. So when you go to the upload page, you can, either create a new project here, or you can select from your recent projects to add your to add new samples or new analysis to a recent project, or you create a new one here. Let us now create a new one. I will just call it demo, and then the 15th January. Here, like I said before, concerning the data privacy, you can choose between creating a public project or a private project. I would always recommend to create a private project. In the end, you can always choose, go back and open your private project for the public, but you can never go back. If you choose here to do a public project, you can never go back to make it private again. So just make sure to always use the private project here. You can give a short description, for example, workshop January 2025, and then you can create a new project. So one thing regarding selecting your projects here, if you want to add new samples to a recent project, I would always recommend to choose the right project here, because the project selection will affect the pipeline and the database version. So, for example, if you have an ongoing project since two months or something, and you've uploaded data already, you have to make sure that the same database version is used within the whole project. Otherwise, you can't compare your results. So make sure to add the data always to the right project here. So then we can go on to the analyze type. Like I said before, there are 4 different analyze types, which can be selected here. But unfortunately, there are only 2 for the nanopore sequencing. So here, for example, the metagenomics can be done with nanopore sequencing, and the SARS-CoV-2 consensus genome can also be done with nanopore sequencing. The other 2 are only for Illumina, so short read sequencing. But maybe they will change it in the future, which would be really good for us nanopore users. So let us now select the metagenomics analysis type and choose nanopore sequencing. We are using the base caller setting here. We can choose. We are now going to use the first base calling. So we can go on to selecting our files. You can either do that in dragging and drop your files here, or just click to get them from a folder from your computer. So let me now give you an example. This is how the files are saved after receiving them from your from the sequencing, from your Nanopore sequencing device, you get the different folders. The most important ones are the ones which the FASTQ pass files. And like I said before, your samples are barcoded. So each sample has one barcode, and we will just focus on one sample. So we just go to barcode 1 and we select all of the barcodes. A bit quickly. Like this. And then you upload it. And it might take, I don't know, a little while. Yeah, okay, this was really quick because we don't have so many files. You have to make sure to not upload too much, otherwise you can't press continue here because it's just too much for one analysis run. And I think we can click continue here now. Yeah, so this pipeline mostly accepts FASTQ files, which makes it really easy for us because we get the raw FASTQ files anyway. And when we go to the metadata part here, the initiative itself recommends to give you to add much data as possible here. It depends on your needs. I would recommend to just do the required or to just add the required data here, which is just those 6 differentiations here. But if you want to add more information to your project, you can always add this via this plus sign here. Yeah, for example, if you have your host sex, if you have the known organism, you can also add it here. For now, let us choose here because we had only culture here. So the host organism is a part which is really important if you want to make sure that if you choose one host here, the DNA of your host will be removed from the analyzing. So, for example, if you have a sample from, I don't know, mice, you can choose it here, and the software itself will remove all sequences which belong to the mice genome. The same for the human, mosquito, whatever you have. So, for example, if we only have culture like we have here, we can choose the last bit where the host will not be subtracted because, yeah, we don't have any host DNA here. Then you can choose the sample type. Like I said before, this is only culture here. So I would use cultured isolates. You can say if it's a water control or not. In our case, it's a no, so we just keep it like that. You can choose between the nucleotypes, so you can choose DNA or RNA. Then you have to add the collection date. I would keep it as broad as possible. So I don't know, it's 2025. And then I would just add like any date here, and collection location. Yeah, again, if you have concerns regarding the data privacy, I would just add the random location as possible. So here I just add Germany for now. Okay, then you can press continue. Okay, here you only see like a review of the data you've added. If you would have added a host organism here, it would be listed here, and yeah, the program itself would take care that you get all the data aligning to that specific host will be removed. Then you have to agree to the data privacy policy. And here it's really important to not close this page before it's the uploads are completed. Otherwise, you have to repeat everything again. And then you can go to the project when it's completed. And you can see here the status of your uploaded samples. So for example, here you can see it's running. It takes around, I would say 10 to 15 minutes for one barcode to be analyzed. So while it's analyzing, you can't see any results yet. You only see the characteristics you have added to your specific sample. So I will now show you an example because it's not, yeah, we don't want to wait for this 10 minutes. It's the same barcode I just showed you. So here you can see it's completed already because it was run yesterday. You can click on it to see the results. And here you can see the highest alignment results for your specific sample. So let me now guide you through this setup. So we've got the taxon of the species which are found or which are aligning to your samples. Here it is, I think the one of the most important things is this one, which is the bases per million. So the number of bases which in all reach aligning to a given taxon. So here you can choose how the whole setup is sorted. So if you choose, for example, bottom down, you can see that here the Acinetobacter is the one which is the most bases aligned to from your samples. Then you've got the total bases here. You've got the read count here. You've got the contig, and then you've got the average percent identity of alignments to NCBI, and you've got the length of your sequence, and you've got the e-value. And with the e-value, it's really important that this one is the expected value, so the average expected value of alignments against the databases, so either the NT, which is the nucleotide database, or the NR, which is the database on amino acid level. And the e-value represents the number of matches with similar quality one would expect to see by random chance. So this parameter provides a measure of randomness. The lower the e-value, the lower the probability to getting a match or an alignment by random chance. So the closer the e-value is to 0, the better. So this is the setup you get. Basically, you can also click on the species which is the most likely in this case. For example, here you can see the different species, and you can also see which species level is most likely to be or to align to your sample. You can also see if it's a known pathogen, then it has a flag like this or this red dot here. You can download it. You can also click on it and you get some basic information on the microbe or the pathogen here. You can also do a coverage visualization, so you can see the covers here again and a bit more precise. One thing I would also recommend is if you decided for yourself that this one is the most likely solution for your sample or aligning with your sample, you can always blast this against the NCBI database again, just to make sure or to confirm this result. So here, like Prakash said before, this you can also use some filters. For example, the you can set the following thresholds. You can use use different categories. So for example, we've got a culture here, so we want only to have the bacterial data, but you can also choose between other microbes or categories here. Then you can always set other thresholds filters. For example, the base pairs, like Prakash said before, shouldn't be underneath 1000. So you can also use this as a filter. So all of this is 100. The ones with less than 1000 base pairs are filtered out of this data here. You can choose between the read specificity. Then you can always annotate the results yourself, so you can mark it for just for your records if it's a hit, if it's not a hit, or if it's inconclusive. And you can also choose to have only the ones which are known pathogenic here displayed. And then you can clear all filters to have it back to your original analysis. The whole data can be downloaded easily here with this button here, or shared with other users, depending if you are working in the group. And I think this is mainly it. You can see also again your sample details, which you've added before. Maybe one more thing which is important: when you go to the pipeline spot here, you can see one category here, bases remaining. There you can see like at what point you lose bases. So for example, the validate input is just like a normal input, which should be always 100%. But then when it comes to the quality filter, the program itself is adding a filter to your data. And if you are losing a lot of bases here, so we've lost 10%, which is okay, but it shouldn't be more than that, because then it means that you might have low quality sequences, and you are losing a lot of your data already doing this filter action. Then if you have a host filter in there, you can also use some more data here, because of course the host data is subtracted from the whole data. And human data is always subtracted, just for also data insurance reasons. So you cannot analyze any human genomes with this database. Yes, I think that's about it for now. Let me stop sharing my screen, and I'm happy to answer any questions if there are any. Okay, if there are no questions, I can really recommend this database if you have to do like some basic analysis. Otherwise, if you want, yes, please.
Okay. Thank you so much for the presentation. I saw that when you display the identity, percentage of identities, I saw that so many bacteria have the highest score of more than 90%.
Just give me a second.
Yes. I'm wondering how do we be confident that when we choose, like here, I think we have 94% for the second bacteria, Escherichia coli. Yeah, because we have already 93. I think we have so many species that have a high score.
Yes.
How?
That's a really good question. It always depends on the way you analyze it. That you have to be sure that the data is really correct, or the results are correct. So at this point, it is quite yeah, not so clear to say. Maybe you've got a sample with both species inside of your sample, because you can see you always have to keep in mind all of those different categories. So it's really important to see the percentage identity. You're absolutely correct, but that at the same time, you always have to keep in mind the basis per million and also the weed length, which is really important, because like if you have really short weeds, like for example the ones here with 6 just 6 weeds, you have to say that it's really unlikely that this is a correct finding or a correct alignment to your sample. So you have to really keep in mind all of these different characteristics. And as the first one here has the most bases per million and 331 reads aligning to this species, it's most likely that this one is the correct one. But yeah, if you want to make sure that it's the same or that this is the correct result, I would always recommend to blast it against the NCBI again to confirm this result. I think that's a thing you should always keep in your mind, that it's not a clear finding here. You always have to analyze it yourself, and you always have to keep in mind your clinical question behind that. So it's always highly depending on the sample you've got, what is realistic that might be in there, because sometimes you're finding in your analysis which just can't be true or is not relevant in your case. So you don't consider that as relevant here. But it's still depending on your analysis here. So it's also a bit okay. Okay, I think we can then go on to the next platform, which is the Genius platform, which is a little bit more comprehensive for maybe also bioinformatic users. And my colleague Arianna Ceruti is going to present that to you.
Okay, thanks, Ria. Can everybody hear me? Okay, I'm going to share my screen once again. All right. Okay, so you've heard about Epitomy and CZID. So those are very simple to use, handy platforms that need the internet to work. And I'm going to present to you Genius. And the nice thing about it is that you can use it offline. And it's a little more customizable. And so, if you're working in the field in remote settings where you don't have connectivity, or you're sitting in a train in Germany going from east to west, you will also have serious connectivity problems. So you need something that allows you to do your work and your analysis offline. So for that, we regularly use Genius. And the main advantages about the platform are, the offline use that I just mentioned. It's fully customizable with like basic bioinformatic tools to analyze all kinds of sequencing data. I'm going to show you a few examples in a minute. And very important, it's a bioinformatic tool, but it's made for non-programming scientists. So you don't have to be able to write code. You don't have to write code. You really just have to, let's say, click and collect. Okay, so this is really nice for veterinarians like us, for example, who are not trained bioinformaticians, who don't have to write code, and you still want to get your customized, let's say, data analysis workflows. And another really nice thing, personally, I think about the platform is that you have a really active user community, which is really handy if you have very specific questions. Everybody's really active, posting and commenting, so that's something you can use, and I totally recommend. There's a variety of tools that you can use with the program, with the software. I'm going to show you, let's say, 3 different ones that we use normally, that are really simple, that are really useful. So one is primer design, blast, and alignment mapping, and de novo assembly, which is something that's really popular, right? So you want to be able to use that. You can also use all other tools like, building a phylogenetic tree, so all kinds of different stuff. And I'm going to show you the software in a minute, so you can have an inside look. Let me share the software. Okay. All right. So the first time I opened this, I was a little, let's say, intimidated by the whole thing, but it's actually really simple. It's really intuitive. On the left side, you have your folders. Yeah, just like on your laptop, the same thing. On the upper side, you have your, let's say, working field, where you can click whatever you want to work on. And on the lower bottom side, you have, let's say, the visualization of whatever you're clicking on. Yeah, so you have these 3 fields that allow you to do whatever you want to do. So the first step is, of course, importing your data. Very simple, just like in any other program, just import your folders. The folders essentially are the sequencing data that you get out of your device. Yeah, so the barcodes that Ria mentioned in the beginning, you will see them here. And if you zoom into your barcodes, you will see many different, let's say, IDs, letters, and so on. And if you zoom into it, you will see your DNA. Yeah, so this is the DNA, the reads of your DNA of a specific sample. Yeah, it's really nice. Finally, you can see some colors, not only text. So it's it's really nice to actually see the base pairs that you want to work on. Here on the right side, you can see the basic parameters. You can change whatever you want to change. You can annotate. You can display it in a way that it's easier for you. So you can change the color coding. You can base quality. You can see how is the GC content of your sequences, the melting temperature, so all kinds of things. So you can just read out here on the right side. I'm not going to go through all of them because it's really depends on what you're looking for. Okay. So this is the basic setup. You have your folders, you have your, let's say, the sequencing data and the visualized sequencing data, so all of the texts that you want to work on. And one of the, let's say, more simple things I can use also without having the subscription, the paid version of it, the trial version, you can do, let's say, basic analysis like importing sequences from NCBI, and you can design primers, for example, for PCR or for isothermal amplifications. You can just import it from NCBI. Very simple. You just copy paste the annotation, the accession number, and here, for example, what I did is I imported a sequence from African swine fever virus, the capsid protein, which is used for genotyping of the virus. And you can see it here, all of the sequence annotated so you know what you're looking at. And what I wanted to do in this case is design primers for an assay, for a molecular test assay. I just went into the whole sequence. You can see it here. It tells you how many base pairs you have, how many gaps, how is the annotation. And in this case, I just chose a region that fits my primer, let's say, characteristics that I want to work on. You can just select, for example, in this case, 32 base pairs of a primer that I will use for my African swine fever region, for a primer, a probe in the middle that fits the probe needs, and a reverse prime. So this is super simple. You don't need a prime version. You can just select sequences, look into it, and maybe look for primers, for example. Very, very common thing to do in our field, right? So this is the first thing, very simple. And then, of course, you have tons of other stuff you can do. Here on the upper side, you can see all of the workflows. It's like I mentioned, you don't have to code anything. You can just click on the thing that you want to do. Yeah, so very, very basic. Yeah, so one thing I want to show you as a tool that's very frequently used is the BLAST. BLAST means basic local alignment search tool. Essentially, it's just you look for similarities in the sequences that you're clicking on. Yeah, so very simple. You want to see, is there a match or not? Okay. In this case, for example, we tested mosquito samples. So whole body mosquitoes from a certain Greek island. We wanted to see, okay, what species is it? Can I say, based on the DNA that I find in the sample, which species of mosquito it is, right? So in this case, you click on your reads. Yeah, this is the reads that came out of your sequencing device for this specific sample. Okay. Here on the upper left side, you can see BLAST. It's singled out because it's very commonly used. And then there's different ways to do this. So Genius obviously wants to make your life as easy as possible. So it has created, let's say, the basic databases that you can use for your similarity search. In this case, for example, 16S ribosome RNA, if you want to do bacteria and bacteria, or you can do environmental samples, you can take all of the NCBI genome. So they prepared for you the databases that you can use on your data set. But then, of course, depending on your aims and your goals and whatever you're looking for, you want to customize your databases that you want to use for your BLAST search. And in my case, of course, I wanted to see, okay, which mosquito species is it? So I needed to create my own database based on the species that I want to screen for, right? To do that, you just need to download your sequences from NCBI that you're interested in and pack it into a folder and create a database. 3 clicks, super simple, and then you can work on it. So in this case, I call it Mosquito ID 2024. I click on BLAST, custom BLAST, so the ones that I created. And then here, of course, you can adjust your algorithm as much as you want. I use the template, the basic setup, but of course you can use different algorithms. You have to decide on your own what you want to use. I use the basic setup, which is totally enough for my purposes. Okay, I click on OK, and it's gonna do the BLAST run. So it will look for similarities between my DNA in my barcode from my mosquito and the sequences from the database, from the mosquito database. I'm just going to stop it because it will take a while, and I'm going to show you the results. So in this case, this is what the Genius software will tell you. Oh, here I have tons of similarities with Culex pipiens molestus. Culex pipiens is, some of you might know, is a very common mosquito. And in this case, the DNA from this barcode, from this sample, matches with Culex pipiens. So I can say with a certain amount of probability that this is a Culex pipiens mosquito. Yeah, very simple, 4 clicks, and Genius will tell you what it finds. You can do the same thing, for example, with another BLAST, custom BLAST that I did for looking for, let's say, the host IDs for the blood meals that a mosquito might have, right? And in this case, I did the same thing. I blasted my raw data, yeah, from the sequencing. I blasted it against another customized database, which I called I have a lot, host ID. Okay, it was the same thing. It will look for similarities, and it will tell you, oh, here I have 2 hits with, in this case, it's a sheep waterfowl. Okay, so this is a BLAST. You can customize it according to your needs. Very useful tool, very popular, used a lot. Another tool I want to show you is the mapping. Mapping can be really useful if you want to say if your sample matches a certain reference strain. Yeah, you want to be sure if and where exactly your sequences attach to a reference sequence. So in this case, I can show you an example based on influenza virus. So here again, you can see all of the basic, the raw reads from my sample in a color coded. And then here on the upper side, you can select your mapping tool. Workflows, map to reference. Yeah. So this is again the basic the setup. You can, change the algorithm tool according to your needs. You can tell them, okay, what kind of software did I use? Do I want to trim some of my sequences because I use primers on it? So many different things that you can click on, and you can, of course, again, select the references, the reference sequences that you want to map your sequence on. In my case, we sequence an unknown influenza virus, and we wanted to say, we want to discover which H and N type it is. So which influenza virus is it? Is it H9N2? Is it H5N1? Clinically also relevant questions. So we wanted to see which one it is. You click on the workflow. It will, take a while to actually give you the results. And in the end, it will look like this. Here you can see the different, let's say, align or mapped reads to the different genes of influenza, which I used in my database. So here is the NS gene, the MP gene, and NA gene 9, PA, PBA, PB2, H15. So as you can see here, it's the 8 genes that influenza viruses have, and the reads that actually align to those genes. In our case, it aligned with H15 and N9. So I can say this is most probably H15 N9 influenza virus. Okay, very handy. 4 clicks, and you will have a first set of results. You can do mapping to a reference, but of course, some of you might be interested in doing de novo assembly. So they reconstitute all of the genome of your sample after it has been trimmed, after it has been, let's say, cut to those different reads, right? So this is a very, very common, let's say, scientific question. You can do the same thing. Here you can see there's a workflow you can click on. We'll have to select. Let me just find here. You will have to select the reads that you want to assemble. First of all, I mapped it to the specific gene. And then what I did a de novo assembly, click on those reads, and it will give me the context that Prakash mentioned before. So you basically, the de novo assembly tool looks for overlaps between your sequences and recreates a longer sequence, a longer context. And in doing so, it will get you in the end the full genome. Yeah. So you go from reads to context to scaffolds to the full genome. That's let's say the different steps going towards your full assembly. And in my case, for example, here you can see the different contexts for the NP gene. And in the end, it will give you a full, let's say, consensus sequence for your NP gene from this influenza sample. So this is, let's say, the crash course on a few of the tools that we use regularly. As I mentioned, you can use all kinds of different things. As you can see here, you can just, let's say, play. It's your playground as a scientist to play with your data according to your needs, according to what you're looking for. Very handy. There's tons of posts and YouTube tutorials that you can use. Yeah, very easy to use. We use it regularly. I really like it. Once you get into the whole flow, let's say, of the interface, you can just go ahead and play. And yeah, that's my part. If anybody has questions, feel free. Otherwise, we can also have a talk via email, depending on your questions. And yeah, feel free to ask questions, and feel free to actually use the softwares to get familiar with it. They're really really useful. And yeah, I hope we could give you a little introduction on those 3 different platforms. We're not getting paid by any of these softwares. We just use it a lot, and we think they're actually very helpful. So yeah, thank you so much, and go ahead if anybody has a burning question. Yeah, everybody is, I think.
Hello, Ari.
Hi, Andy. Hi.
How are you?
Hi, nice to see you.
Yeah, a nice presentation. Thank you for everything, you, Ari, Rea, and Prakash. You talked a little bit about the possibility of doing primers designing for isothermal amplification. Did you ever try it with this software?
Yes, we actually did that a lot. It's not automized, so for PCR you can just click, okay, give me PCR primers. There's other online softwares that you might know that do that as well. For RPA primers, there's not such a software, so you have to design them on your own based on the basic RPA characteristics as a primer needs. You still have to look for them on your own based on the sequence. So there's not a let's say ready to use RPA primer design click on Genius, but you can still use it. Yeah, for PCR, PCR is very common. Everybody, designs PCR primers all the time. So there's automated, let's say, pipelines for RPA. Not yet, but maybe we might do that on our own. Maybe that's an idea.
Perfect, thank you.
Yeah, thanks. All right. If that's it, thank you so much, everybody, for coming. It's almost 2 hours, but we kept the time, the timeline. Do we have any other closing remarks, Ria?
Yeah, no, thank you very much to all of you. Yeah, it was really nice to have so many listeners here. I think in between we were more than 90 people joining, so that's really, really nice. Like I wrote in the chat, we recorded the whole meeting, so we can share the records with every one of you if you want. And yeah, thank you very much for your attendance. Have a good day, everyone.
All right, thank you.
Bye-bye. Bye.
Have a good day.
Bye-bye. Thank you for your good presentation.
Bye-bye.
Bye. Bye, everybody.